So I first learned about scAnt through the publication that came out last year, and I was very, very excited because it complemented a project that I'm on in the United States called Lightning Bug. And so they do somewhat similar things in that they take, can everybody unmute? I mean, mute themselves. They're similar in that they both provide these 3D models of images for us domain scientists to work on. The other thing, and what really started Lightning Bug, was the ability to take images of labels on the specimens and then create a virtual label, go through OCR and parse it out. And so correct me if I'm wrong, David or Fabian, but so that's something that scAnt really doesn't deal with, right? Yeah. So scAnt focuses more on 3D modeling of insects, and I just think it's really, really exciting because one of the challenges that we have in the United States is, for us domain scientists, is to engage computer scientists and engineers. It's like they're typically not interested in our problem, or they just don't know that we exist. And so I think one way to get around this is to identify people all over the world that are champions of some kind of technology that we can put into place. And I think scAnt is an absolutely wonderful example of that. And with that, I'm going to now turn it over. So you can start sharing, and I'll also make you guys. I'm not
sure if I can share my screen yet.
Look for the little green button that says share screen at the bottom.
Okay. It says host disabled participant screen sharing.
Oh, you're right. Okay. I forgot to hit share. Okay, now do it.
Let's give this a try. And please let me know when the full screen works on your end, because I'm not entirely sure. Perfect.
It works
great.
All right,
then David is going to do the introduction of the system, and I'm going to take over at a later point in time.
Okay, sorry. Hi, everyone. I hope you can hear me and see the presentation. Thanks very much for the kind introduction and for the invitation to speak here, Neil. We're very excited to present this to this audience and see what they actually have to say, both critically and positively, about the system scAnt, which we published just a few months earlier this year. Okay, so I thought in the beginning I should point out or say a little bit about the background, because we are neither what Neil called domain scientists, nor are we really necessarily engineers or technology people. We sort of humbly sit somewhere between the interface of physics and biology. We at Imperial College at the Department of Bioengineering. I lead the evolutionary biomechanics laboratory, and we currently do research in 3 different fields. We look at the biomechanics of plant-eating insects. We look at how insects are able to climb on surfaces and stick as well as unstick quickly. And we also start to look more and more into the energetics and mechanics of insect locomotion. Now, I'm not saying this because I'm going to tell you about these research fields, because it's not why you're here. I'm saying this to stress at the beginning that what we're going to talk about is actually not what we usually do. Right? So this was sort of a side product of our research that we thought or felt in interaction speaking to others, that maybe other people might actually be interested in being able to use this for something else. So it will hopefully become a bit clearer when Fabi's taking over later why we actually built this device and what we want to do with it in our research. But we are a research group that as such neither works on digitization, nor necessarily on imaging, nor necessarily on technology, but in a different field. But in that field, we wanted to do something, and Fabi will explain what that is later, that led us to build this device, scAnt. And I thought in the beginning, although we now figured out that the volume might not work, and some people might have seen this video on Twitter, I just show a quick video that demonstrates sort of from the device to what we're actually trying to do with it. And then I talk about the different aspects a bit more in detail. And I'm going to play this. And can you hear the sound of this? You can? Okay, cool.
Actually, David, it's pretty quiet. I hear something garbled in the background. David?
Is it still not better, the volume?
No, it doesn't sound at all. Nobody can hear anything.
Oh, okay, so I thought I asked some people that said they can hear, but okay, so people couldn't hear. Okay, so I mean, it's basically explaining a process. Let me try and play it, and I turn off the video, and I turn off the sound of the video. So it was basically just in the beginning here, it's providing motivation that plenty of you can hear me now, Neil, right? This is all fine.
Yes.
Okay, cool. Just to confirm, there are large natural history collections. Plenty of these mean that if you want to access the specimen, you have to go there. If you go there, you have to take out the specimen physically. That comes as risk to damage them. And people take images, but these are 2D. And to overcome this problem and provide 3D images, we've developed a system that we call scAnt that can help people to make 3D models. And you see here basically a video of the process. You have an insect specimen in a lightning dome. You have a camera rail that takes focus stacked images. And you can then take these images, or you can control the hardware with this graphical user interface. You produce these focus stacks. You can merge them into one EDOF image, and then you can make 3D models from them. And that's the first part of the process. So you can make these textured and color realistic or semi-realistic color realistic models. We can also do more. We can rig these models. So we can give them bones. We can make them move more or less realistically. We can also place them in artificial environments. So these are all 3D models, and they're all in 3D generated environments. We can do that with several different species. And we can then try and teach computers things to do with these species. And that's what sort of we actually want to do with these models. And that's why we build a system. And that's what Fabian is going to talk about at the end. Here are just a few examples. We can track animals. We can pose estimate with the data that we can generate. We can start estimating weight automatically from video recordings, so in particular polymorphous insects. And these are some of the things that we are trying to do with scAnt, but I'm sure other people have plenty of other ideas of how this could be useful. Okay, so there's no replaying. I didn't want that. I want to go further. Okay, so really what scAnt does is it helps with the process of generating 3D models from 2D images. And in general, there are two main steps to this process. The first one is that if you have an object that you want a 3D model of, you have to take images of that object from multiple perspectives. That can either mean you move the camera around the object, or you move the object around and have the camera static. And then from these images, and that's the process that is actually called photogrammetry, you can generate these sparse point clouds. So you locate the position in 3D space of different parts of the body, and you connect all these points and you create a textured mesh. Now, scAnt does in principle nothing with the second part of this process. That's done with either open source or commercial software. That's entirely up to the user. But it does help with the first part of this process, namely the generation of these images. And to help with that process, we've developed two parts of scAnt. One is purely hardware, and you've seen this in the video briefly already. And the other one is a graphical user interface that allows the control of this hardware and also some image processing steps. And I'm going to briefly talk you through both the hardware, the graphical user interface, and the image processing steps. Okay. So these are just a couple of images and technical drawings of the hardware. So you see that in principle, you have this round lightning dome. In the middle of that dome is a sample holder that sits on a 2D gimbal, which can be rotated about 2 axes. So that allows us to rotate the specimen and take images from different perspectives. There's a camera that sits on a slider, and that slider moves the camera closer or further away. So we move the focal plane through the depth of the specimen, and then we generate multiple images, and in the end can generate one single image with only pixels that are in focus from that particular perspective. There's a second slide that shows this in more detail here. Top drawing also with some of the technical components, the motors, the control, the steppers, etc. And in the bottom, you see the process maybe a bit more clearly illustrated. The camera can move from left to right. The gimbal can rotate about 2 axes. There's a stop that makes sure that the camera never runs into the lightning dome. And you also on top see the lightning dome with a mounted specimen in the middle. There's a slit so that the gimbal can move. There are LEDs mounted inside the dome so that we have flat lighting and consistent lighting across different orientations or for different images, which is very important for the subsequent process. And that is, if you will, in essence, the hardware. Now, this hardware can be controlled with a graphical user interface, and here's just a screenshot of that graphical user interface. It has a few different components. On the top left, you see that you can control camera settings. That includes classic things as exposure time, gain, gamma level, balance ratios for red and blue, black levels, etc. You can see that it also automatically shows you overexposed areas on the specimen. These are these red dots. Shows you red color information in the bottom right. You can use auto exposure features. You can correct the highlighting exposure, etc. And that allows you to set the light settings, if you will. And the bottom left is a panel that allows you to control the relevant motor parts. So that, for one, in the very bottom is the focus axis. So that's what moves the camera. And then there are the two rotational axes. You can define start and end points, and these will in principle depend on the size of the specimen. They will also depend on the resolution that you want. So for the rotation, you can define how many rotation steps you want to take an image of, which allows a more detailed or less detailed reconstruction at the cost of computational time and, of course, final model quality. The top right is, if you will, housekeeping. So you specify where the resulting images are saved. You specify certain settings for the images that you can also save and load later for other scans. And you can activate simultaneous processing of the recorded images, which I will speak about in a second. In the bottom right, then, it's just an info panel that tells you about the progress of the scan. And this is, in essence, the graphical user interface. Now, the ability to stack images already is essential for the subsequent photogrammetry process. So we do a couple of things in terms of image processing to produce input that can be used in a large number of photogrammetry softwares. We have these captured images. They have to be aligned. Then we have to restrict them to images that contain only pixels that are in focus, or contain no pixels that are out of focus, I should say correctly. We do that with these focus masks at standard image processing techniques, and we can select only images that have relevant pixels. We then have these extended depth of field images, but we still need to remove the background from these to have them usable in photogrammetry software. And we do that again in a sequence of steps. Essentially, what we want to do is we want to select the largest shapes. For that, we do, again, some image processing in terms of the Gaussian blur and the Klarheit process. And then we detect this shape with a random forest-based outline detection. That detects the largest shape and is all connected. So it also assigns white pixels, if you will, to areas that don't belong to the specimen. That's shown in the middle of these three images. So the areas that are highlighted in red are not specimen areas. And we can still select them with image processing techniques, and then remove them in a subsequent step. And then we have a mask that we can apply to the extended depth of field image. And we end up with a masked image as input for photogrammetry software. You can do this at any point after the actual physical scanning or physical taking of pictures, or you can do it while you take it, which saves time, but requires a minimum amount of computational power. Okay, you then have image processing, and you then take these images and feed them into the photogrammetry software of your choice. We're not going to talk about this because this is not a contribution that we really make. We worked a lot with Measurum because we like working with open source software, but other software is available and can be used with these images. In the bottom, you see a couple of examples. These are images of the 3D models, and at the top, you see a slider that goes between an actual fully rendered model, which then looks relatively nice because you can have realistic shade and lighting, etc., and the neat raw model, if you will, without texture and color information superimposed here of a bug that we work with. Okay, so you can make these models, and of course, what many people in this call will be interested in, or so I assume, is what quality these models actually have. Before I talk about that, we do a few post-processing steps, although strictly none of these are necessary depending on the model quality that you're after. Most of the insects that we scan are pinned, so we can remove pinning artifacts, which you see here in the bottom. This is a manual step, so we haven't automated this. It takes, therefore, a period of time, depending on how experienced users are with software such as Blender, but it's possible, and you can remove it. You will still, with a keen eye, see where the pin was, so it's, of course, not possible to perfectly model what would have been there if the pin wasn't there, but you can close these surfaces and have them of similar appearance. What we can also do with these models, which will be interesting in some of the things that we show later, is that we can rig them, which means that we assign a skeleton. So one advantage that we have here is that arthropods have an exoskeleton, which can be suitably modeled as a rigid or a segment sequences of rigid bodies that are linked together. So we can define these rigid bodies and joints and then link them together. And that allows us to pose these specimens. So you see on the very left here, an example of a stick insect, which we scanned that had a curved abdomen and legs that have all sorts of funky positions. And because we can assign rigid bodies to these and then pose them freely, we can do things like straighten the abdomen and bring the legs in a natural position. So that provides us with some advantage because we can, in principle, move these specimens around after we actually scan them. And the last thing, which I think is also important to stress, is that there are, of course, some problems which I will talk about in detail later, but one very common one is thin and/or transparent structures. The key difficulty here is to assign whether a pixel belongs to the top or bottom of something. If something is very thin, that can easily be confused, or indeed to identify it if it's transparent. So in particular, wings are challenging for free photogrammetry software, and some commercial software are some more powerful algorithms, but they cost money. We included one relatively simple, if not perfect, way of dealing with this problem, and that is that we basically take wings from masked images and reproject them onto the model, which then means that you get a relatively realistic appearance, but the wings are simplified as 2D planar structures. So where there are wings that have significantly corrugation or curvature, that gets lost. In some cases, that is probably fine and works reasonably well, and maybe this example is one. In other cases, that will be a significant simplification and might yield less realistic models, but that's an ongoing problem. And well, we are hopeful, I guess, that the next 5 years of development and code for photogrammetry software will make further improvements there, because this is nothing that has to do with the models that are produced. This is a problem in the meshing process itself. Okay, in terms of quality, there are a few things that people might be interested in. We can, for example, talk about minimum size. The smallest model that we produced here is a woodlouse on the right. So you see a 5mm scale bar. It's reasonably small, and it still produces reasonably good results. In principle, the smallest size that you can reconstruct depends on the optics that are used. So if you use a higher magnification lens, smaller sizes might be possible at higher resolution, but that determines the resolution. The larger size is limited by the physical dimensions of the setup, but in principle, it can be scaled up if you want to scan larger specimen. In terms of color, here's an example of a quite, well, if you will, complex, colorful specimen, and color seems to be reproduced nicely, in part due to the favorable lighting conditions inside the dome. You might be interested in something like mesh detail and things that can be reproduced. And for this, there's an example in the bottom here of a Camponotus giga specimen that shows you the mesh detail even at small scale. So the mesh is still detailed. You see here that at that scale of the claws, so we're looking into micrometric dimensions, of course, the anatomy is no longer perfect. Right? So I'm going to say something about resolution later, but here you still see that claws are there, but they don't necessarily reflect very accurately the actual shape. And the last thing that might be interesting is something like topography at smaller scales. And here's an example of actually one commercial and one free meshing software, the free software at the top to the commercial software in the bottom. And you see that both of them actually do a reasonably good job at retaining small scale topography on the specimen. But the commercial software does a slightly better job at retaining some of these fine scale bumps. So these are just a few insights into the quality of the models. Now, in particular for this audience that might be interested in using this device, or well, potentially using this device for research, I thought we should briefly say that there are still some meaningful limitations. There's limitations on specimen size. I briefly noted this. The small size is to some extent just controlled by optics. So if you have multiple lenses available, that is addressable. The large size is currently limited by the dimensions of the scanner. So if you build a scaled up version, larger sizes are possible. But with the system that's currently built, I think it's between 5mm and 8cm is the maximum size. The second one is accuracy, and it's difficult to actually give a good figure to this because there's multiple ways in which one can think about it. The main problem is that what is the ground truth you're comparing to? So you need something with very well-known dimensions to say something about accuracy. We reconstructed gauge blocks that have very high precision tolerances in manufacturing, and they suggested that in these instances we have a reconstruction accuracy of about 10 micron. Now, that applies if the structures are relatively bulky and this is a height difference that we can resolve. Whether we would necessarily say that we can reconstruct every single 10 micron structure is a different question, because it also depends on the number of pixels that belong to these structures. So it's relatively difficult to give a good number on accuracy for these models. We're still thinking about how we could best do this, because of course it's important, but the best thing that we could come up with in terms of the publication we put out was this reconstruction of these gauge blocks. Having said this, a classic example of where this becomes difficult, and we're quite possibly at worse resolution, are high aspect ratio structures. So something that is much longer in 1 or 2 dimensions than in 2 or in the 3rd dimension. Examples of this are proboscis that often are fragmented. Example of this are wings that are planar. And here it can become difficult to pick up all the pixels correctly or to have the reconstruction process accurate. And then we can still produce good looking models with some manual labor, but of course that's really not ideal if you want to do this at a larger scale where you want to minimize any manual labor that might be required. What is perhaps surprisingly difficult are smooth surfaces. So you maybe seen that stick insect model, which comes up very nicely because of its topography. But if we look at very smooth surfaces, which then are reflective, for example, in other ants, the texture doesn't necessarily look entirely realistic because that changes with orientation. The appearance changes slightly. So we get problems in the reconstruction again, and it looks a bit more clay-like as a texture. So these are still problems. There are a few things that we suggest in our manuscript what can be done to address this, but it's not ideal. Transparency is difficult. I talked about this with the wings. And then I think for many people in this audience, particularly important is time. And there are actually 3 time components that we think are relevant. There's one that I call the setup time, which is basically if we go and make a scan, how long is it from taking the specimen, putting it in to starting the scan? And for an experienced user, that is anywhere between 10 and 30 minutes. That depends to some extent on whether you have to change the image settings or not. The scan time itself, which is, if you will, machine occupation time. That's how long the machine will be used to make a scan, and that's between 2 and 5 hours. Depends a bit on whether you do simultaneous image processing or not. And then last but not least, the reconstruction time, which depends, of course, on the computer and the detail of the model, varies to some extent between 3 and 12 hours. Now, the reconstruction time is, again, something that we have relatively little control over. That's computer power and photogrammetry algorithms. We help with some extent of that because we provide some input scripts for original orientation of camera positions, etc. But I think the main things where we can do something in the future are setup time, not necessarily, but scan time. And I will say something about this in a second. And of course, also in terms of accessibility, it still costs some money to make it, right? I mean, it's, I think 180 quid is roughly the figure at the moment, but it's still some money. So not everyone has that amount of money. And it still requires some equipment to have access to. And although it's rapid prototyping, and many institutions will have access to a 3D printer and a laser cutter, not all will. So if we really want to make this accessible as broadly as possible, that is something that stands in the way of that. And then last but not least, if you want to build the device yourself, and some people have, I think we're now at what, 6 rebuilds or something maybe?
So there's 4 published on Sketchfab, but far more from people that have just community-wise rebuilt it.
So of the order between 5 and 10, say. So some people have already built it, but these are keen builders that have some previous experience. I'm not sure, maybe some are even here in the call, but you still need to solder, you still need to figure out how to put these things together. So there's still a bit of effort required to actually build this device yourself, although all the information that is available is in principle there. Needed information. Yeah, and there's still some restrictions in terms of camera needs. It's quite flexible. I mean, you can use right now DSLRs and a number of machine learning cameras, but it's not that you can use any camera that you possibly want yet, although that might hopefully happen or to some more extent in the future. And that's also what I want to say next. We are currently working on a proof of concept grant to bring this further and to hopefully make it cheaper, faster, better. And I just want to briefly highlight a few of the things that we're planning or hoping to achieve. We want to reduce the costs. Our aim is really to reduce the costs in half again. So the material costs, excluding the optics. The optics will always be the most expensive part of the system, unless you use very cheap cameras, of course. But if you want to have sensible model quality, the optics will be the most expensive part. But one of the things we want to do is we want to replace the stepper motors with the 3D printer board. There's a couple of other things that hopefully make it cheaper. We want to reduce time. We think that the main point with which we can address this is to improve the communication between the camera and the computer and to increase stability. And we hope that we can roughly half the scan time. We want to increase flexibility, add support for more cameras, provide design options for smaller and larger specimen. And we want to increase or improve accessibility, simplify the manufacturing process and the assembly process, and develop a scan kit similar to 3D printer kits that exist, for example, for Prusa, so that you would receive the parts and just have to do the assembly. And hopefully a simpler assembly process without soldering it, etc. We are committed to keeping scAnt open, so we have no interest in closing any of this off. We will continue to share all the information that is needed to build your own system. If you are in a position, and Neil has kindly done so already, to write a letter of support for this grant that outlines why this might be useful for you, then we would very much appreciate if you send that to the email address below. And that will hopefully help us secure this money to make sure that we can develop this further, because we can't do it in our normal research grants or normal research time because it's out of our usual remit. But we recognize that there's an interest in the community, or there seems to be an interest in the community. So we hope to bring this further. Okay, so that's roughly what I wanted to say briefly on scAnt. We think it's affordable, accessible, and performant. It's relatively cheap. It uses generic electronic components. It has support for a large number of cameras. Manufacturing relies entirely on rapid prototyping. You can now use it with the help, actually, and that's an example of why accessibility is nice. One person who built their own device added support for Mac operating systems, so we now have all common operating systems in place. You can find all the information on classic sites. Code is on GitHub and documentation. Parts list and drawings are on Thingiverse. Model examples are on Sketchfab, and we think it's relatively reasonably performant. You can go from a pin specimen to a full 3D model within about 10 hours. And that's all I wanted to say about the system scAnt, and I'm going to hand over to Fabi for the next 10 to 15 minutes for what we actually do with scAnt, which might also give some people ideas of where to go with the system. Enjoy.
All right, thank you very much for the wonderful introduction of the system. And let's now actually have a look at what kind of things we can do with the system now that everything is in place. Now, first of all, we have quite the range in terms of the different sizes that we could potentially throw into a scanner. And obviously, the models at this point are still relatively static. And while for applications such as we want to do take measurements from these scans, those can be 1D, 2D, or even 3D. If we care for volume measurements, for examples, this will be perfectly fine to have a static model. The same goes for general conservation efforts. So if it's just about digitizing these specimens, we could in theory stop here. But as David kind of said earlier, this was not the primary objective that we had when first designing the scanner. Now I'm going to say something very, very outlandish in just a second, but this is important in order to understand why it's actually relatively easy to now bring some life back into these models. And that is that all insects are the same. Now I know this is a very wild claim, and not everyone will agree here, but it also only matters in the ways that it matters. And that is we have more or less the same building blocks to bring these models to life, meaning we have a hat, we have a thorax, we have an abdomen, the legs are segmented into the parts that we kind of show here on the side. That means that within our models, like David teased earlier a bit, we can actually define a 3D rig, meaning that we say where the joints and where the virtual bones, if you so will, are located inside our model, and then tell the 3D software of our choice, in our case that would be Blender, which vertices, so which parts of the model to move in accordance with what bone. This is exactly what's happening here. So we start out with a texture mesh, that can be either from one of the other insects we've shown, or specifically here with some of our leafcutter ants. Then we turn that into a rigged and posable mesh. So here you can kind of see, I hope the resolution is high enough, that we actually define all these joints and virtual bones inside of that model. And once that is taken care of, you can basically take our previously static mesh and now pose it however we want. And now one way to bring life into this is to use so-called markerless 3D motion capture. And we have built a system for that in our laboratory. This one was built by Hendrik Beck. And what this basically does is we have a largely automated recording setup here with 5 synchronized cameras that all capture at the same time. And then we can have insects walk across this platform here. Now, the interesting thing about that is that we can also change the level of inclination that we have in order to study how those would affect the locomotion of these insects. But first and foremost, at least for this purpose here, what we can now do is that we move away from just having this in a 2D setup and bring it back to our 3D models. And this works in a couple of different steps here. The first one being, you might have heard about Deep Lab Cut, a great software in order to do so-called 2D pose estimation without any markers. So rather than having to obstruct your animal while it's walking around, you can define essentially landmarks on your model where you want the machine learning model to re-identify it. So here we have all of these wonderful 2D pose estimations after producing lots of training data that the system can now relatively robustly identify, at least as long as everything happens on that confined walking platform. And now with using any pose, we can first synchronize all these cameras and then calibrate those cameras. So we also have an idea where these points are actually located in 3D space. And by then projecting the underlying skeleton that we have defined back on these points, we can see how this would kind of then shape out to be. Now, as long as it's just in this form here, it doesn't tell us too much about also the orientation or rotation of some of the virtual bones that we have defined in here, but we can basically directly transfer that onto our 3D models. So here, once again, we have the rigged mesh of a Songaia inexpectata, second insta, I believe. And here is some of the motion capture data that we actually used from our 3D markerless motion capture system that is directly transferred onto the models here. But in my opinion, this is also just the beginning of all the cool things that we can do with it. because I personally care even more about investigating behavior of social insects, more specifically leafcutter ants. So one thing that also this graphic here displays quite well is that we have highly polymorphic colonies, meaning that all the workers are of vastly different sizes, and depending on how big they are, they will also do different things within the colony. The only exception there might be the queen. That is, she obviously has a very different role for other reasons, but the polymorphic workforce that we kind of see here do lots of different things depending on certain biotic and abiotic factors. And those factors basically influence at what size ants will do what thing. So the smaller ones tend to be gardeners. The average range of workers that we have will be foragers, so they will do the actual leaf cutting. And the very biggest ones will be the ones primarily occupied with colony defense. But sadly, all of these rules are not actually set in stone that much. So depending on where these ants are, depending on the feeding state of the colony, all of these rules are to some degree interchangeable. So if we now want to study at very high detail this size-dependent division of labor, we need to start using automated tools, because at some point it just becomes impossible to keep track of them. Here's a very good example of how that might look once we're looking at the actual colonies. You have hundreds, if not thousands, of individuals in a single view, and it becomes very difficult to tell them apart because the colonies are very large, you have high levels of occlusion, and you have a frequently changing environment. But what we essentially want is very, very complex information. So we have to use a multitude of different tools for this very specific research task. So we need to know their location, means we need to perform detection. We need to identify the same individual between different frames, meaning we also have to do tracking. We need to classify their size because we have different size classes. And we also want to some degree, infer what tasks they're currently performing. That means action recognition. And ideally, and this makes it specifically difficult, is that we also want to use all of these tools outside of our laboratory. Now, as many of you probably know, especially if you have some sort of technical background, is that deep learning more and more becomes a standard in this toolbox of a behavioral scientist in the animal world. But that really only works if we have sufficiently variable data sets. And I've done the work. I've done the the big hand labeling, meaning I've annotated over 10,000 frames at this point of ants in different environments, all of them walking around. But the problem with that is, well, if I just have these 10,000 examples and all of them are captured under laboratory conditions, well, how well do these networks that we train to then take care of all these tasks that I just mentioned, also in more challenging environments? Now, one thing we can do is so-called data augmentation. So we take these original annotated samples and just basically change their appearance without changing their meaning. But that still really only gives us limited variability because the background will still stay the same. While the exposure might change, the lighting situation itself is also somewhat the same. We can also not really influence the proximity between different individuals. And this is where producing synthetic data sets comes into play. And this is really where the whole idea of scAnt started, meaning that once we have these digital models, we can be a lot more variable with the domains and the environments that we can simulate to produce the variability that we need. Now, other people have done this before in rather different scenarios, because this is already quite popular for the automotive industry. You see it in human pose estimation applications, robotics applications. Most of the time, wherever there is enough money to build such a system, you will find someone using synthetic data. So if we would also want to do this for our applications now and keep all of this cheap, that also means that we need to develop our own data generator that can make use of the rigged models that we have produced. And this is exactly what we have set out to do. Now, this specific generator I'm developing jointly with the Pocket Dimension in Munich. And when we think about what this generator has to do, it's a couple of different things. And we need to think about it as what is the domain or the environment that we need to simulate, and what are the animals that we need to simulate. So when setting up this generator, we first need to understand how many samples do we need. So how many images do we need to throw out at the very end? We need to define the number of individuals that we want to have in there, control the density, so the proximity between individuals, and set up domain variation, so what kind of noise and everything we want to throw on top of it. And also, we need to set up the individual variation. Now, for these leafcutter ants, for example, which vary in size and also to some degree in the texture, varying texture, material, and scale are things that we do automatically. So we can simulate these huge colonies with all of them as independent actors in order to build machine learning data sets. Now, this is a simplification of how the process works, but just to give you a brief overview of how these samples then could be generated. The first thing we do is we set the domain size, so how much space the colony can occupy for that specific image. We randomize the pose of all the individuals that are in there. Then we randomize the environment. That can be as simple as just loading a background texture, all the way to using more complex lighting environments and different levels of occlusion. We then place and rotate the camera randomly within that scene. We add noise to the sample, and then we finally render the sample and export all the information that we could possibly want from it. Now, here are just a few of the examples of how the samples actually look like that come out of such a data generator. Some of them look relatively realistic, like this forest floor setting here. Others can be more described as some weird abstract art, but all of them do the trick of bringing more variability into the data sets or being perfectly annotated. And now here's something very interesting. We actually started training different detector networks only using synthetic data in order to identify ants in our scenarios that we have here. So it works relatively well if everything is relatively controlled. So we have nice lighting conditions. There's not too many workers at the same time, or at least a manageable amount. And they occlude each other as much as ants normally do. The same thing also works surprisingly well when we have texture backgrounds. And now something to kind of keep attention here to is down here, we have the same performance based only on real data, meaning that the networks that we have trained have actually been trained on these tens of thousands of images that I have annotated myself. And even though this example that we see right here has never seen a single real ant, so it has only been trained on synthetic data, it outperforms the networks trained on real data in every single scenario that we presented. So the same thing is also true if the scene itself is overexposed with lots of motion blur or underexposed with flickering. And we can see that a few issues certainly exist here, but in every single case, our synthetic data has shown to be of more variable quality than the real data was that it was ever able to annotate in the laboratory. And now something to keep in mind here is obviously these are still relatively controlled examples, but the same thing can still be applied if we kind of move it into the field. So here we have some handheld footage that I've just recorded with a cell phone to kind of show that the same techniques absolutely work when the scene is a lot more difficult to analyze. So we have lots of motion blur, there's lots of animals at the same time in the same scene, and the detector still does a reasonably great job at picking all of them up. Same thing we can see here again with even more camera movements and lots of individuals there at the same time. Not sure how great the frame rate is, but this does actually run in real time on relatively modest hardware. But now let's take a step back and actually think about what are the possible applications that we have now that we can generate these data sets. Well, first of all, we can define unique identities in our colonies. So all of these ants can be re-identified between different images, meaning tracking is an opportunity. We have semantic information at a pixel level, which means that we can train networks such as mask RCNN, if you've heard about those, which basically traces the exact outline of every single object that you have, which can help a lot if you need to identify different species in the wild or they're just on challenging backgrounds. We get perfect 2D and 3D joint locations of every single object in there, meaning that for every single generated sample, we know the 2D pose, basically in pixel space with the segmentation map, but we also know where all of these points are actually located in 3D space. So we might be able to do much more complex pose estimation than we could ever do with just hand annotated 2D data. And if we're very, very ambitious, and this is something that we haven't really tackled yet, is that we can also produce depth maps. So if we're interested in how far every single pixel that we see in our scene is away from the camera, which can help with things such as size estimation, when we want to know, well, is this just a scaling thing, or is this ant actually this big or this small? This is some extra information that we get with every single sample that we generate. So now to quickly summarize. scAnt makes digitizing animals, especially insects or arthropods in a broader sense, much more accessible. The pipeline that we have developed is applicable to basically all insects that we throw at it, at least the ones that we can rig with the skeleton that we have defined. Synthetic data does surpass traditional data augmentation in all the cases that we have at least identified so far. The synthetic data also produces much more robust deep learning than could be done with hand-annotated data alone. And it really does minimize the labeling effort while maximizing the information that we get out of the samples that we can produce at the end of the day. So thanks very much for all of your attention. I appreciate that this was a longer presentation. So hi from everyone from the Evolutionary Biomechanics group. These are our research sponsors that we have on the side down there. So please feel free to ask us all the questions in the world. We can't wait to hear them.
He's muted. Neil's muted.
Neil, I think you're muted. No.